clicked. Hey everyone, it's Clara and I'm back with another chat with one of my science buddies. So a few weeks ago I did a recording with Izzy, talking mostly about equality and diversity work and stuff like that. But this series is more focusing on the science, and Izzy is a scientist, so I want to find out what it is she does and learn from her and learn basically. So that's where we're at. So what we're going to do is we'll cut over to the video in a minute. I will say that I'll put some links down below that Izzy has shared, so feel free to follow her and look at the links that she shares, and also feel free to sort of like and subscribe and all the YouTube-y stuff, because numbers are good. So I'll be back in a second with Izzy. Hi Izzy. Oh, I've muted you. Right, there we go.
Hi.
How are you doing? You okay?
Good, yeah, how are you?
Yeah, great. Thank you so much for sort of after me already doing one chaotic chat with you. Thank you for coming back and doing another one. That's really cool.
Pleasure.
So do you want to introduce yourself? Where do you work? And sort of very overviewy of anything that you want to talk about, and you really introduce yourself.
Okay, so my name is Izzy Jayasinghe. I work in the University of Sheffield as a Senior Research Fellow. I'm a UKRI Future Leader Fellow, looking at using microscopy in sort of new and exciting ways of looking at sort of life in the sort of the tiniest spatial scales. As in terms of who I am, I'm a trans woman and a lesbian, and I guess I'm not from this part of the world. So I was born in Sri Lanka and grew up quite a lot in New Zealand, and I've sort of worked in 3 countries in the last sort of 10 years, sort of navigating a career in science. So in kind of like a nutshell, that's who I am.
Yeah, perfect. The irony, I've only spoken to, you're the 3rd person I've spoken to today and the 2nd person from Sri Lanka. So that's, yeah, I mean, like I say, I've not talked to many people today, but there you go. So, yeah, I mean, you said that you work in microscopy. Now, I work with microscopy as well for looking at materials, and I use scanning electron microscopes and things like that. You've actually been sort of, when we talk, you're building up your own microscope. So what sort of microscopes are these? How do they, very rough, how they work, and why it is that you need these new microscopes? Why are you building them? What's wrong with the ones that you get from, Toys R Us?
There's always something new to be built. It's the bottom line. But in terms of the need, I think if I start with the need for it, I trained as a biologist, so we were looking at biological samples. And one of the characteristic things, especially if you're looking at the biology inside sort of the human body or like mammalian animal cells and tissues, one of the striking issues is that they intrinsically lack contrast. So they all look quite white or very pale. And if you wanted to look at the building blocks inside it, they all look colorless or very similar in color. So there's been a need to kind of build microscopy tools that will add contrast. And that was a feature in all of the microscopy that's been done over the decades, going back more than 100 years. Every time you need to visualize something biological, you need to add a stain. Now, there are techniques that use intrinsic sort of color or spectral properties to get that contrast, but by and large, you need to add a stain to it to visualize some aspect of it. And it's true for electron microscopy or optical microscopy or other things. So I work in optical microscopy, which means that we tend to use stains that give it color, and the specific types that we use fluorescent dyes, which make them glow. And then, the techniques that we develop from there is kind of exploiting that fluorescence, that kind of glow, to better see structures.
So that's awesome. So what sort of level are you looking at? Are you looking at the sort of the cell level, the atom level? Where are you going to? How far are you going?
So it's been a bit of a journey. I would say around the time that I started my PhD, which was going back about 12 years or 13 years, the scale that we could look into with optical techniques was sort of hundreds of nanometers. So 250 nanometers was sort of the barrier, and we call it the diffraction limit, because we just didn't have the lenses and the tools to go beyond that in terms of resolution. But 10 years on, we are hitting resolution optically around 1 nanometer. And that's been achieved with different approaches, different methods, and they broadly we call them super resolution microscopy, and super resolution microscopy is sort of the overarching theme of my research.
I find it amazing that you're getting that sort of resolution with an optical microscope. I mean, I'm using scanning electron microscopes for that sort of level, but all my materials are metals or pieces of ceramic. It's really easy to do. As soon as you introduce water or something, it's a very different prospect, which is why we need so many different techniques, right?
Yeah, I mean, I have to clarify that the rules of physics still haven't changed. We've gotten better at building lenses, so there are lenses that can, like in their straightforward use, can reach resolution sort of below 200 nanometers easily. But a lot of the finer improvements, sort of getting down to single-digit nanometers, is a combination of optical imaging and computational analysis. So it's not, strictly speaking, genuinely true to say that you can use a lens to see things optically in the scale of 1 nanometer. There are often reconstructions based on the way we use the optical device.
Yeah, no, that's a good point. I also love the idea that you're dyeing all these cells and they're all having a little rave, like with the little fluorescent things. But that's just the way my mind goes, really. One thing that does strike me, so obviously you're talking about the human body. So, I mean, are these microscopes ones that you're using in people, or are you using them on samples sort of that have been removed? How are you doing this? You stick a microscope up down someone's throat.
Yeah. So the work that I do in my team are definitely not ones that can be directly focused on to humans or animals. So what we tend to do is we take biopsies, so little chunks of tissue or little samples from people or animals or from other organisms, and then you prepare it on a slide, and we put that slide on a microscope. But the same principles have been used to look directly into living or conscious organisms. The most common one is a head-mounted fluorescence microscope that can image directly into the brain of a conscious mouse or a rat. I don't think that equivalent one has been done into the human, but there are fluorescence microscopes that can image right through the cranium, the skull, into the brain.
That's so cool. That's so cool. And how did you get into this? So you said your first degree was biology. So what was your kind of career path? What was your journey?
I've kind of gone in and out of biology So when I was in high school, my favorite subject was physics, and I won a scholarship to go to university to study science on the back of a physics project that I did. Um, and then when I got to university, I didn't really believe in my math skills. I thought my math skills were quite weak. So I picked one that was easy on the maths, which was biology. And I quickly learned that the interesting biology happens in the interface of biology with quantitative fields. Um, and so I kind of found a research group that was working on biophysics. So they were kind of imaging cells, and they were making measurements from those images, and then they were making deductions about how the physical processes that drive life in those cells. For example, things like diffusion, things like electrical signaling worked. So they were still embedded in physics. And that group that I joined, they were interested in cells of the heart, and they were using optical microscopy to make those measurements. And over the years, my PhD was in physiology, technically, but since then I've gradually become more interested in the tool and the question.
It's funny how it changes, isn't it? And I think that's the nice thing. You're a little bit similar to me in the I'm a material scientist, and so I started in engineering, but then there's the physics, there's the chemistry. It's kind of materials is the hybrid science, and it's a little bit similar to what you're doing. It's like there's a bit of biology, but there's a lot of physics as well, and it's a real mix.
Yeah,
and I think that makes it really interesting.
Yeah, and also you can't really pigeonhole yourself into one discipline. I mean, some people do, and that's their career choice. I think of it all as just science, and if I need to learn something from chemistry, I go ahead and do that.
Yeah.
Always sort of. Well, I mean, it's a way of challenging oneself, but I think it makes life and work interesting if you are prepared to learn new things.
I completely agree, and I've been doing the same thing. I'm sort of currently learning as well. Interesting, actually. So I count myself. I do research, but I don't lecture. Do you also lecture? Do you teach?
I do. So teaching has been a part of my career at various stages. So when I was a PhD student, I also worked as a tutor in the department. I think these days they're called as sort of teaching fellows. So I was kind of like an equivalent of a junior teaching fellow part-time. But then more recently, when I kind of became an independent researcher, that was on the agreement that I would take on a certain amount of the department's teaching. So I was teaching mostly biology, so physiology, but then I was also teaching some courses and modules where it's more technically focused, sort of. And the aim was not just to just deliver more and more content on biology, but to kind of instills the scientific skills that are transferable when it comes to the students. So, to be able to write a good report, to be able to plot graphs that are scientifically accessible and clear, to be able to analyze data and do presentations and things like that.
Yeah, that's really good. It just when you were speaking, it just made me think, because I was whatever they call it, doing some teaching when I was doing my PhD, but I was my PhD was in material science, and I was lecturing in the teaching in the engineering, because that was my degree. That's what I did. So the material side has always been more practical, and since I've been in sort of materials groups, and especially now at Oxford, I don't feel like I don't feel like I have the background necessarily to be able to do it. But of late, with putting together certain videos and learning different subjects, I'm like, hey, hang on, maybe I can. I was worried I'd fried my brain, basically. I thought that I'd frazzled it.
I guess the thing to remember is that teaching or lecturing or tutoring, they are forms of science communication. So the main difference is that you are now working within a certain structure, a certain program, to a certain plan that fits into a bigger sort of program of teaching. But essentially, the basics are still the same. The basics are to sort of engage and captivate your audience and kind of make that topic interesting and accessible, and give the students the tools to learn however much they want to learn.
Yeah. Do you enjoy do you prefer the teaching side? Do you prefer the research side? Do you like a mixture of both?
It varies from time to time. There are, to be really honest. So the aspects of teaching that I really enjoy is standing up in front of the class and kind of introducing a topic that the students have not been exposed to before, and really exploring it and having a conversation with them about it, and observing how they are engaging with it and how they really run with the ideas. And that's the fun part of teaching. The aspect of teaching that is less enjoyable, and I think as a kind of a sector higher education needs to evolve on, is how we assess teaching or how we assess the learning. And as a teacher who has been now do the marking, it is quite backbreaking and kind of soul destroying work to mark 80, 100 reports of the same type again and again and again, and things that are written in a very prescribed way. And there's a big field on teaching pedagogies in higher education, and there's a thing called the Bloom's hierarchy of learning, and at the very bottom are things like the learning of the facts, the knowledge, and the details. At the highest levels of learning, you're talking about being able to synthesize something new. And I think at the moment, most universities dwell a lot on the facts and the rote learning, and not really reaching for that understanding and being able to synthesize something new. And I think, I would enjoy that part of that marking, for example, if we transitioned into higher levels of learning as a sector. And I think there's some work to be done on that.
Yeah, I completely agree. I'm someone that has to learn by doing. It's as simple as that. So if I've done an experiment, even when I'm reading, because it's relating to something that I'm specifically doing, I pick up on things and it just stays. Whereas if I'm just reading, I can do quite well in exams, but it's gone as soon as the exam's finished.
Yeah.
I don't learn things. Whereas if I'm actually working on things practically, it's
like especially it's a bit like learning a new technique and then applying it. After you've done that experiment a few times using a new microscopy method or a new manufacturing method, you have a much more sort of cohesive and three-dimensional sort of understanding of it than if you, for example, had to write an essay or a paragraph in an exam.
I think we don't necessarily get learning right in universities. There's a lot of people that are forced to teach who are good researchers but don't know how to teach. And like you say, when it comes to exam time or reports time or whatever, I see how busy our heroes are, and it's kind of glad I'm not in it.
Yeah, it's also the economics that don't really work with that philosophy.
Yeah.
So you know, there's an ongoing debate about the value of the money that students pay to universities to get their learning, and how best to how efficiently to really deliver that teaching. And I think especially in times like this pandemic, when it's very difficult to learn in person in a group or in a lab, it's very difficult to deliver those sort of deeper levels of understanding. And it's a real shame.
Yeah. I mean, yeah, obviously we're learning a lot now and having to change how we do things, but I also think that there will be positives. I think for we're starting to realize that people who do struggle to get into lectures for one reason or another, and there's plenty of reasons, I think we're starting to see that there are more tools and we can change how we do things. But also, I do like, especially with labs and things like that, you need to be hands-on. You can't do a virtual lab unless it's software, maybe. I don't know. It's
yeah. I mean, I think even what you're doing through your YouTube channel is actually kind of ahead of that curve, because what we've seen in universities is academics being asked to suddenly get on to video tools and use animations and sort of short videos to really drill into the details of what they're teaching. And I think this is going to be the new norm. And it doesn't, just because you're not sort of teaching, or recording these videos for a specific class, doesn't mean that this is not the new way. So I think there's lots that the universities are learning from kind of the YouTube generation.
Wow, that's interesting. I've wanted to do it for years and years, animating the science. I thought that it'd always be really well. I mean, I'd love to actually work with professional animators and really, really break it down. And there are some YouTube channels that do that really well. And I feel like mine are basic. I mean, they are basic. I'm not, they are basic, but it doesn't matter as long as you can still get the points across. And that's, and that's what I hope we can do. And
I think just looking at science YouTube, absolutely there are some channels that are really good at using animations or other kind of visual aids, but there are also channels that have been successful that are not so reliant on graphics. So I think it's not a single formula. It's the whole package that you deliver.
Absolutely. And the truth is that two different presenters could have really similar style, but there's something that just clicks with one of them and it doesn't click with the other. And that's what we've got to remember. I don't think it's difficult in universities. I don't think it's a weakness if someone's like, I can't learn from you. I can get an overview, but with this person, I understand it deeper. And I don't, we shouldn't be looking at that as a weakness. We should be looking at what the difference is then and sort of analysing it and using that to improve our teaching. But our lecturers, most of them, a lot of people, they're researchers who have to teach, and it means that they feel that they're doing the teaching because they have to do it. And we need to give people more time and more training, but universities, whether they will.
I think it's personally I find that good teachers, good science communicators are uncorrelated with in terms of how well they're doing in research. Going back to the pre-YouTube era when I was an undergraduate, there were some amazing lecturers. So there was a lecturer who taught us in cancer biology, who who was a really captivating lecturer and drew cartoons. So on the whiteboard, he would fill the whiteboard so that we'd have about 3 or 4 whiteboards in the room, and he would fill them with cartoons, and they're immaculate. And he would he would anthropomorphize some of the things. Like, for example, if you're talking about immune cells that attack cancer cells, he would draw them as little army soldiers and so on. So it was really accessible. And he was a teaching fellow in the university. But then there was also another professor who was a really, a real superstar. And he was a captivating person to listen to. Just, he didn't have any extra talents or aids, but as a speaker, he was quite captivating. So, I think, you don't have to be a superstar in scientific research to be a good teacher, but at the same time, you could be. So it just depends on what your passions are and what you're interested in.
Absolutely. Yeah, no, I mean, I've had amazing lecturers who were great researchers, and I've had terrible ones in either direction. I mean, that's always the case. Going back to your research, so you mentioned so we are currently in the sort of pandemic lockdown era, which means that we're changing everything about how we work. I mean, obviously your day-to-day job is in the lab, right? So how are you doing at the moment? Are you back in the lab? Are you getting towards getting back in the lab?
I'm in a really precarious situation between kind of. Well, I ended my previous job in a different university, and I started in my new position during the lockdown.
Yeah,
it's a strange time to start a new job. Yep. And the issue has been that all my kit, all my microscopes in that previous lab still locked down, so I can't really begin my experiments properly in the new setup. So it's a constant sort of communication process of working out when best to, get access to that equipment and then go and set it up.
Yeah. So are the labs in your new building, in your new university, are they starting to open up now? Are they?
Yeah, they are opening up in limited capacity. The emphasis is really to keep the density of people down. Yep. Because for SARS-CoV-2, the virus, I think the biggest risk is enclosed spaces that are not very well ventilated, and people kind of going in and out of those spaces or being asked to share those spaces. And so there's still an encouragement to work from home as much as possible. But in terms of what I do, I would say about 50% of that work is image analysis and writing software, yeah. and about 50% are kind of experimental. So there's no shortage of data at the moment for me from the pre-lockdown times that I can analyze on my computer at home.
That's really good. Yeah, for me, I mean, I need to make new samples, I need to make new films, and then I need to analyze them. A lot of what I do is, if I'm doing the research, it's in the lab.
Yeah. Have you been going in? Lately?
No, the building, I did the risk assessments for the labs I think they went in about 2 weeks ago, and they've been signed off most of the way up, but they're, as you say, trying to open the labs one at a time to reduce the sort of impact on the thing. So I wasn't sure. Like last week, I thought I'd be in the building last week, and this week I thought I was going to be in the building this week. So it's like, okay, maybe I'll be in next week, and I probably will. But then again, I seem to remember saying that on Monday as well that I'll be in next week. So I know that we've got some postgraduate and PhD researchers who are really keen to get in, and they sort of take priority. So I've got to get in and get the kit up and running for them, and then I can sort of. I mean, my research at the moment is very much on the side, so I'm there to facilitate their research, and hopefully I can do some of my own, which I have. But it's difficult. It's a difficult time for everyone, and obviously, I mean, we had a postdoc, so I feel bad for you. We had a postdoc arrive, and they moved to the UK, and they arrived a week before lockdown, and
what a time to arrive!
Yeah, and I was like, I can't teach you to use the equipment because I know full well that it's going to be it's going to be a month before you're back in the lab and using it, and you'll have forgotten. So I'll teach you again, and there wasn't anything that I could really do in that time. So I was like, you need to read, and then we'll come back in a month and we'll teach you properly. That was back when we thought it was going to be a month.
Yeah, I think it really hammers home the reality that a lot of scientific work that's been done relies on skills that are passed on from one person to the next. And in a situation like this where it's the air that we breathe that transmits the disease, where teaching those sort of skills from one person to the other is really difficult, and that's the real barrier for science sort of progressing in this time.
That's one of the two big issues that I've realized. One is that I will have to teach him how to use potentially dangerous equipment, complicated equipment, and so I've got to be able to do that and do it safely. Once they know how to use it, they can just get on and use it on their own. It's fine. But the other thing is that we're also sort of like you. I mean, not the same scale, but we're also building a new microscope, and so I had all the parts arrive before lockdown, and sometimes you need two people to put things together and to test them and everything. So,
and you need time in the lab. Like building a microscope is not something that you can do to a schedule easily. You need lots of time and time to not just build but to validate the tool.
Yeah, exactly. I think, absolutely. It is unusual, and I'm curious how it's going to impact research sort of down the line, especially with grants and stuff like that. So you've got you got a really amazing fellowship, right, in terms of prestige, and you're a UKRI Future Leader. I mean, that's brilliant. Tell us about this. What it means. What it is.
Yeah, I mean, I think it was sort of applying the right scheme at the right time, but essentially what it is that it's a fellowship that's designed to support people who are not necessarily fully established as researchers or independent researchers. But also don't fit neatly into a specific box. So I mean, as I said, I trained as a biologist, but a lot of what I do these days is sort of embedded in physics, chemistry, and engineering. And over the last 4 or 5 years, I've struggled to get funded for that kind of interdisciplinary work because I would apply to a very specific funder that focuses on medicine or biology or heart disease or something like that, and the full gravity of that proposal doesn't always reach them. And when this fellowship scheme came along, it was the first time that the proposals were being assessed for being really good all-rounders. So it wasn't just about your science; it's about your capacity to lead, and demonstrating that you can, for example, put together a network of researchers that will work together on the same project. It's things like having a plan on, like if you're building something, having a plan commercialize that technology. So I think it was the sort of scheme that was built for the kind of researcher that I am.
Yeah.
But it was also it's a prestigious fellowship because each fellowship lasts 7 years.
Wow.
So they give you funding upfront for 4 years, and then you have to kind of apply for the remaining funding. But essentially, you're a fellow for 7 years, and it gives you that time to build a research program that no other single grant can do. And it means that you can really dream big and be really ambitious, and also at the same time grow as a person. So by the time you finished your fellowship, you should know what your direction is, whether you're well established by then as a senior academic in the department, or whether you want to go and establish a company, or go into politics or whatever. And the fellowship scheme is not just necessarily for science or engineering. It's a range of people. So from humanities, from disciplines like psychology, medicine, and so on. So it's a cohort that's very diverse. It's a bit like hitting the jackpot to get one of those.
And I mean, you said the right place, the right scheme, the right time, but also you got it because of the merit of your academic work and your knowledge, and you're very sort of like, oh yeah, think, very, what's the word, modest, and but you got it because you've got the skills, and this sort of scheme is exactly for people like you who are, but they're struggling to get the grants because they're not always understood. So that's amazing.
Yeah, it takes a bit of practice to get there, and I mean, for some people perhaps it comes naturally. I'm not sure, but for me, I had a few close failures. So I had interviewed for other fellowships, and at the very final stage, I would get knocked back. And I think those failures have really stretched me as an academic, and sort of it's pushed me to sort of refine the way I write and the way I communicate science. It's painful, it's exhausting, and I think even in the previous conversations we've talked about how the landscape for getting funded is not flat. There are some real issues around equity for various groups who are disadvantaged in the process. But I think that landscape may be shifting, and the UKRI, for once, I know are definitely conscious about this issue. If not, taking some action towards it. Only time will tell whether their actions and what they're doing is going to be effective in the long term.
Yeah, I mean, I was in a weird position because the PhD I went to was already funded, then the postdoc and the visiting posts were all already funded, so I was already sort of 8 years in before I had to apply for anything, and that's when I was really making my decision on transitioning. So I wasn't publishing, and my mental health wasn't good. So the last 5 years I've been working, I've been able to do research because I managed to get a job, a lab management job, which they were very happy to let me do research as well. That I've got a really amazing group, but now I think I'd have to sort of, I need those career break fellowships or whatever, because it affects us in different ways, and there's a lot of reasons why we're not applying for grants or why we're not getting them even.
I mean, I think there needs to be a broader conversation about how we as the scientific community are doing science, because there's definitely a big part of our community who don't have the time and the support to write grant applications. But and it really comes down to balancing out, what are they happy doing and what do they want to do. And writing competitive applications is a good strategy if you want to do something that you can't currently do. And at the same time, the funders need to look after the area of science that are sustainable, or basically that are ongoing based on what is fundable, as a given. So I think those are the two aspects of science that I think we as the community need to support. So as a community, we shouldn't kind of stop people from going for funding for something ambitious that they want to do at the same time, while taking away the science that they like doing. It's quite challenging thing to challenging balance to achieve, I think.
Absolutely. And actually, I don't know, I mean, it'd be nice to just talk a little bit more about the sort of microscopes and the research and stuff. So, I mean, so like you say, in order to be competitive with grants, you've got to show that it's commercial, or that that it'll be used, it's practical. I think Blue Sky's research is really, really important. I was talking to Andrew Prinsep, and that's a lot of what he's doing, which is great. But you can't always get that funded.
That's right.
So what is the use of the microscopes, the techniques that you're developing? What are you using them for? You said it was to be able to see at a different resolution, but why do we need to?
So my work is sort of driven by a mix of blue sky research and sort of commercially aimed developments. So the blue sky research sort of is aimed at understanding how individual proteins interact inside the cell and work together to kind of sustain life. And they kind of link to diseases and to understanding things like heart disease. But the biologist in me is interested in knowing how they work. So that is the blue sky aspect of that research. And the microscopes that I build allow me to do things like map those proteins in space. So in a cell, in a three-dimensional volume, I mean, a cell is relatively large. So a mammalian cell, you're looking at about, multiple microns across. But if you look at a single protein, you're now starting to get into single, like nanometers, a few nanometers. Some of the proteins that I look at are a bit larger. They're about 30 nanometers. So you can see them optically with these techniques individually. And what you can do is you can have a map of where the proteins are and conduct virtual experiments. Once you have that map in a computer, you can run simulations on how they interact and how they produce a signal, or how they drive some kind of response to the cell. In the heart, the cells are muscle cells, so basically these proteins kind of make the cell contract. They shorten physically. So you can actually run those experiments in virtual space if you have those precise maps, which you can't usually do experimentally sometimes, because if you're looking at live imaging of cells while they are doing their thing, you can't really get that level of detail. So sometimes those experiments need to be done in virtual space. So that's the kind of blue sky aspect of it. That's the motivation. The other motivation has been a more practical one, and that is to help biologists, generally speaking, to see structures better. So there's a clear difference between resolving the structure at a resolution of 100 or 200 nanometers compared to resolving it at 1 or 2 nanometers or 10 nanometers, for example. So there's a real race to just sort of improve that resolution, and there are different strategies of getting this. One is a method or a family of methods called Localization Microscopy, and I'll send you a link to be posted below the video of how it works. There's one video made by a colleague of mine that kind of illustrates this point sort of more visually. And that video was a video that he recorded on his mobile phone of the light show at the Eiffel Tower in Paris. So you have these flashing lights on the tower, and when they're flashing, you can kind of make out an outline of the tower, but it's kind of a blurry image. And what you can do is record a video of those flashing lights, and in each frame of that video, you're mathematically, computationally pinpointing where that flash is coming from. And that centroid, that center of each flash, if you then superposition them onto a map, that map suddenly gives you more detail, and you get a much more crisp image of the Eiffel Tower. So I'll link that on the video. You can go and have a look.
Amazing. That's so cool.
And there's a story about it, how they came across it. So there was, in the mid 2000s, around 2005, there were about 4 or 5 groups that were working on this concept. And it was a small number of them actually got recognized for that work. So they got the Nobel Prize in Chemistry in 2014 for it. And there was a researcher called Eric Bedsig, and I'll send a link of one of his talks as well. And he was unemployed when he developed this method.
Wow.
So he had essentially lost his job from his institute, and he had he was talking. I think he was going for a walk with one of his former colleagues, and just generally sort of talking about the great unanswered questions in science, and they kind of arrived at this idea that they hadn't got around to testing. And they went back and approached some colleagues from Harvard, and borrowed some laser equipment, and they set up a microscope in the living room of one of their houses, and did the experiment at home, and went back and wrote a paper in Science.
Wow.
Now he heads up a major research institute in Jamelia Farm. So, interesting story.
This says it all about science, right? Like, this person had lost their job, and as soon as they had a bit more time, maybe they didn't have the pressures, and they were able to think about problems. So this is why, with our researchers, we've got to give them time to think, because that's when the ideas come. That's when often you're relaxed and you're like, oh, what about this, and just try it. And
exactly,
we don't have that. We give people money because we know they're going to get results, and not because they might be able to just experiment and see what happens.
Exactly. And I think these days, especially these days, you have to map every single one of your outputs before you apply for the funding. And so, it's a foregone conclusion. So, you're basically working to a very strict script. There's very little room to go out and explore and innovate in things. And so fellowships really do help with that. The fellowships tend to be more flexible and accommodating of new innovations. And I'm lucky in that aspect. But that's, I think, one of the real issues of contemporary science, that everything has to be done to a budget and to a plan. Yeah.
I got disillusioned, when I was sort of getting towards the end of my postdocs, because I, you know, sort of noticed something and I discussed it through with a few more people, and it's like, wow, this looks like it's a real thing. And when I was trying to write about it, it's like, well, no one's done it before. There's no literature to talk about, so we don't know that it's happening, so you shouldn't be talking about it. I'm like, what? This is, this is like, we might have discovered something and it might have been important.
Yeah.
And it seemed like it was possible, but because there wasn't the confidence, because it hadn't been done before, if it had been done before, they would have been fine with me talking about it. But because it hadn't, like, what?
Yeah.
Oh.
Yeah, you have to really be prepared to explore new opportunities in order to kind of make breakthroughs. There's one example that I can give from my group. So before I got this Future Leader Fellowship, I had a small grant from the Wellcome Trust called the Seed Award. And it was a very specific task that we had to do. And the person that I hired to do that work was a junior postdoc who was straight out of her PhD, and she worked really efficiently and really hard, and kind of achieved all the aims of that grant around about half the time that we expected it to take. And so after that, it was kind of an exploration.
Nice.
And she actually made a mistake one day. So she was trying to use fluorescent nanodiamonds, which you may know about. So these are tiny little diamonds that are produced either synthetically or products of detonation, and we can use them as tags for protein.
Okay.
And they fluoresce, so they glow, and so you can image where the proteins are, right, if you can use them as tags. And this colleague had, just out of the blue, tried some nanodiamonds as labels, and she was using a microscope that I had built. So it was a custom microscope, and things change in a custom microscope, especially if multiple people are using it. And on the day that she was imaging it, she had the wrong filters on, and she discovered this kind of flashing behavior that I described, like in the Eiffel Tower. Wow. Which would not have been seen if the correct filters were on. And then so we wrote a paper about it, and it's now in review with a reasonably high impact journal. Nice. And it was just that, obviously exploration. She was brave enough to try something new. Yeah. But also when we recognized that this was different, it was, we had no explanation for it at the time. Yeah. But we kind of sat with it and we pursued it. It turned into a project, and yeah.
I got an email this morning, so I wrote the spec for a bit of equipment that another group bought or are buying. It's being built, and they were like, and the company were like, so what conditions do you run this at and this at and this at? And I'm like, oh no, we don't. Like, they were wish list things that I put on the equipment just because I wanted to see. Yeah. Like, I've never been able to have them. I've wanted to try them, so I don't know. I haven't got conditions. Might not even use the equipment, but it was basically just a, what do I want to try? I'll put it on this.
There's always an endless list of experiments that you want to do and haven't got the time or the resources to get around to.
Yeah, I really hope I do actually get to use that kit, because I really, like I say, it's not even for me, but I was like, I want to try these things, so we'll see.
Yeah.
We'll see. Yeah, it's funny though, isn't it? Yeah. So I mean, so we've been talking for quite a while now, so I mean, we both know each other because we have been working in various sort of equality and diversity and equity projects.
Yeah.
And so, I mean, are there any projects that you're involved with that you're really happy about and you want to talk about?
I think at the moment, one of the projects that we are both involved in is this Tiger in STEM webinar series that we just launched. And I guess the backstory to that, at least the bit that I'm willing to share, is that the lockdown has seen kind of a real expansion in online events and webinars and online conferences and workshops. And for webinars especially, it has kind of, for me at least, has really brought out the hierarchical nature of academia. So, the people who get invited to give webinars have tended to be, senior professors, often overrepresenting male academics, cis white straight male academics. And you may have seen on social media there's been a lot of discussion about how this is not fair, and how that needs to change. And some of the organizations that have been hosting webinars have been doing that, actively going out of their way to invite female and LGBTQI researchers, and also people who are not cis, so trans and non-binary researchers, to really take the platform and talk about what they do and their experiences. But I have found that is still not adequate. If you look at a webinar series, it is still, in terms of numbers, dominated by people who are not in the margins. And so, we kind of rallied together to create this sort of webinar series to kind of bring a bit more equity. So, not just invite people who are professors and at the top of their fields, but also give the platform to, PhD researchers or really early career postdoctoral and sort of research fellows, and also, showcase that, disciplines like physics are diverse. And just because you don't give them the platform doesn't mean that those groups exist, the groups that are underrepresented. So the Tiger in STEM webinar series is sort of, we kind of put together as a kind of a first step towards changing this attitude. I really hope that it will catch on, and I think by the time this video airs, we will have had our first webinar. I hope it shows everyone that to have a great webinar, what you need is a series of speakers who are really passionate about their work and willing to talk authentically, and it's not about who they are, and it's not about how senior they are or established they are. So that's sort of the thing that's going on for me at the moment.
Yeah, it's so I think it might have started when I put this out, although I might actually try and get it out a little bit earlier, so that we can advertise it. And also, they will be available on the Tiger in STEM YouTube channel as well, which I'll put a link in below. So even if they've happened, you can still watch them, everyone. There are some amazing speakers, like people that I have seen speak, and I am excited to hear them again. Clara Malone, did I be saying names? Oh, we'll be all right. And
I think we went on some of the names.
Yeah, so I'm really excited to watch that. And it's interesting, like you say, it's about making sure that people have got the platform. So I think I know I get nice feedback when I do talk, and people like the way I do presentations and stuff like that. But I'm only talking because I'm, people are actually trying to find someone that's trans, in STEM.
Yeah.
and my scientific career hadn't necessarily achieved what I wanted to achieve, and I'm hoping that I can sort of pull it back a little bit. But I've enjoyed doing the outreach and talking about diversity in STEM, and now I'm starting to talk about the science again. But if you don't give those people the platform, you could lose good people. I've seen some amazing people speak who wouldn't have had a platform if people hadn't made the effort to be more diverse with their lineup.
I have to say that one myth that we're trying to bust is this idea that scientific platforms are, for the work alone, and it has to be dissociated from the person. And that is entirely inaccurate, and it is actually wrong, because who we are and how we do science is deeply embedded in our identities and how we come about. So, I will give an example from myself, because that's the easiest thing to do. I have actively chosen an area of science where I can build my equipment and build the tools that I need for my research, because I trained in a place like New Zealand where scientific research is chronically underfunded, and we were expected to kind of innovate. And we have actually a term called number 8 wire, which relates to the farming wire that they use in farms in New Zealand for fencing particularly. And that, the anecdote is that you can make anything out of number 8 wire. And that, if you look at some of the microscopes that I have built in the past, not necessarily going forward, I have had to have a left field approach, and that was the only way that I was going to get my science done. That was the only way that I could stay competitive in my scientific field. And if anyone says that, well, you can't talk about who you are and where you come from when you talk about your science, that's rubbish. So I think that myth needs to be busted.
Yeah, absolutely. And there are impacts. Of course, there are impacts from talking about it. I had a conversation last night about, the father of eugenics, and it's like, Galston did a lot for maths, but you can't just talk about that without talking about who they are, putting them into context, because that'll push people out.
Yeah,
we don't want, we shouldn't be making people feel uncomfortable.
Yeah, I was watching one of your videos recently where you talked about, your formative years as a trans person, particularly before you transitioned, and how those, how you had to sort of go about life differently. And I have a similar story that when I was a school kid, and I was, quite a gender variant, child. My parents had an approach where they thought that controlling what I read and media that I had access to was sort of limiting, or could kind of control and put me on the right path to being normal. And the strategy that I had, one of the things that my parents actively did was they censored newspapers that they bought. They didn't have access to the internet at home.
But there's a really, really interesting point. So you hear these discussions about nature over nurture, and if you don't give access to young people who are gender variant or have different sexual identities, that they won't become trans or gay or bi. But actually what we're showing is that it doesn't matter whether we've got the knowledge available, we still know, and we will find a way. For me, I just had to wait a long time, which isn't great.
Yeah. I think the take-home message is that you can't hide the truth from people. I think I consider myself lucky to be alive, that it didn't. I didn't let it impact me in a negative way. I probably nearly did, But I think you can't hide the truth from people. So people will seek out the truth, even if they have to go through a massive detour. And so information, especially, you can't withhold information from the public, the people, and they will find it at some stage. And some people, have read stories and or experiences of some people who discover what it means to be transgender, in the really sort of the twilight years of their lives, and that changes them. And however they access that information, um, yeah, you can't control that. You can't control people's destinies.
No, I agree with that. um, yeah, sorry. There was a lot at the end, and it touched me. And yeah, we're like you say, lucky to have you around, and yeah. Is there anything else? I realise I've kept you on a little bit longer. So, is there anything else that you want to touch on? Are you quite happy with where we've got to and everything?
I think a brief thing to say is that how grateful that you're doing this kind of outreach, and also just for being who you are. I guess I have been following your YouTube channel closely in the last sort of couple of months, especially in the lockdown months. And a lot of the things that you are sharing are striking major chords in me, and I'm sure that is true for a lot of LGBTQI colleagues. And I think these are things that need to be said and experiences that need to be shared. And you're doing a great service to that community by being authentic and being who you are, and sharing this openly, and having this discussion. So long may that continue.
Amazing. And it's just, this is my story, my journey, my viewpoints, and my thoughts, and I am not story. This is the point about diversity, right? We all have our different stories, we all have our different backgrounds, and the more stories you hear, the more information you have, the more you can find your path. And I haven't said this yet. Congratulations on the National Diversity nomination. Like, that's amazing. Like,
Thank you.
I'm jealous. I'm not gonna lie. First thought was, God damn it, I wanted that. But I maybe in the future. But I think that's amazing.
I think I'm still in the short list, but if I win it, it'll definitely be a group effort, because I don't know if you've seen the nomination process. It's a lot of writing and a lot of admin to do, and so it's all thanks to all the people who took the time to write nominations for me.
Yeah, I'd looked at it because I didn't know that you were getting nominated, and it's just amazing. I think it's brilliant, and I think you deserve being on the shortlist. It's great. So
thank you. Yeah, it's the colleagues in Tiger in STEM who have really pushed it, and I think you were one of, I think you were the initial colleague who kind of invited me to join Tiger in STEM. And I think that's kind of a real example of what a positive environment can do to an individual, like giving them a platform and giving them a voice. So
I talked a lot about this with Andrew, that what I like about the Tiger in STEM group is that we can get things wrong, and we know that people will understand that we've got it wrong because we don't know, and are willing to listen why we got it wrong, and we're willing to listen why we got it wrong. And there's that learning. Everyone knows that it's like, I don't have the experience of this, so we have other people that we can go to and know that we won't be judged for not knowing, for getting it wrong.
And also the goal of Tiger in STEM is bigger than sort of individual goals. I mean, I have been involved with EDI groups, or equality and diversity inclusion groups, in institutions, where there's always, for the individual, there's always an agenda, there's always a goal.
Yeah.
While in Tiger in STEM, the goal is to make our community better. And I think there's very little that an individual can aim to take away from it other than that big goal. And I think that's what drives us, and that's what makes that community like really, really friendly and collegial.
Yeah. Great. Well, thank you so much. I say I'm lucky to have you as a friend to be able to talk to and learn from, and
likewise.
And I appreciate that you give me time to do things like this because you are a bit
pleasure. It's my pleasure. Yeah.
Cool. Well, with that, thank you so much. I'll flip onto a single screen and say goodbye, but say goodbye to the audience. And
thank you.
I'll put links down below, and follow Izzy on Twitter and stuff like that. Always great to see. Thank you.
Thank you, Clara.
Thanks. Bye. Cool. So that was me chatting with Izzy. What an amazing individual. So I could talk for hours. I really could, but I don't want to take all her time. So yeah, thanks so much for listening. Like I say, there'll be links below to the videos and socials for Izzy that I mentioned, and also feel free to give it a like, a subscribe, share, tell your friends that we're here and we're doing this. That'd be brilliant. And I'll be back with another talk in a few weeks. So bye.